Assalamu alaikum. Good morning, students. I am super excited for this new sort of an online lesson, and I'm really hoping that you will enjoy this lesson as much as I do. Now, before I jump into the lesson, there are a few things I want you to keep in mind. The first thing is I want you to make sure that whilst I'm doing the lesson and you're looking at my presentation, please make sure that you do take notes, okay? And whenever you think that there is an instance where you don't understand, pause the video and then you can see that video again and you can retake the notes, okay? All right, so let's directly jump into the lesson. Now, today our topic is from AS level biology, topic number 4. That's membranes, okay, as you can see on the screen. So what I'm going to do is I'm going to directly jump into the learning outcomes of my today's lesson, and you can take them as the lesson objectives also. So quickly, if you look at the screen, you can see on the screen that the first thing that you will be able to do after today's lesson will be you will be able to outline the functions of membranes in a general sense. Number 2, you will be able to describe and explain, and I really hope you understand the distinction between these glossary terms, description and explanation. Describe and explain the individual components of membranes in relation to their function. We're going to look at 5 of them. And then finally, we are going to look at a process which is known as cell signaling. You will be able to outline the process of cell signaling. Now, just a disclaimer, there are lots and lots of diagrams which I've included in my PowerPoint presentation. And you know, the idea of doing this is because from the exam perspective, there have been instances in the past papers where you have been asked to draw the diagrams. And I will give you an exam style question today also. So please make sure that the fact that I'm giving you so many diagrams does not mean that you're not supposed to draw them. And I'll come to the point where I'll tell you what's the extent to which you need to draw and what you don't need to draw. Okay? All right, so let's directly jump into the first point. All right. Now, I have a list of almost 8 to 9 points which generally outline the functions of membranes, okay? And mind you, when I'm using the word membrane, you need to understand, and you've done that already in topic 1, the cell, may that be an animal cell or a plant cell, its membrane is not just restricted to its surface membrane. You know, there are organelles which are membrane-bound organelles. But generally speaking, these 10 points, I'm going to talk about majority of them. They're going to be about the cell surface membrane, which is actually the boundary. Okay, for both the plants and of course, the plant cell has the outer boundary, which is the cell wall, which we're not concerned with at the moment. So if you look at the first point says that the cell membrane or the cell surface membrane, that's the good way to put, actually that's the correct way to put it. The cell surface membrane acts as a barrier between cytoplasm and the external environment. And you know this fact that the external environment is the tissue fluid. Point number 2. It protects the organelles from damage and from pathogens, okay? As equally, I mean, there are so many organelles we've talked about in the first topic, like mitochondria, chloroplast in the case of plant cells. So all of these organelles are literally protected by this cell surface membrane. And of course, at the same time, it prevents the entry of pathogens. Point number 3. It plays an important role in cell signaling. Now, we're going to talk about this idea in detail, but just to give you a little idea, cell signal, you know, cells, just like human beings, they're social beings, quote unquote, if you like. You know, there is a need in the body for the cells to communicate with one another in case of a threat, in case of something positive, in terms of food, in terms of, you know, maybe any beneficial substance. So there is a way in which these cells, they communicate. Okay, of course, we're going to go into details when we look at this topic, and that's the third objective that we have for today. Okay, so it plays an important role in cell signaling because it has receptors for cell signaling substance, which could be a hormone or a neurotransmitter. Again, we look at details later. Point number 4, it acts as a cell surface antigen by doing cell recognition. Now, this idea is really interesting. And of course, I'll just give you an example, and then we're going to talk about this idea in detail in the topic number 11 to come, that's immunity. And I'm sure that people, this COVID has sparked quite an interest with regards to immunity. So we're going to talk about detail, inshallah, when we do the proteins for this particular topic, okay? In the first slide, I'm just outlining the basic points that outline the functions of membranes. Point number 5, the membranes, they play a role in cell-to-cell adhesion. I mean, think about this. Cells, they're not literally, floating around in your body. They're together with other cells. So they're not glued to each other. There's something that keeps them together. And that is another function of the cell membrane. Point number 6, it is a site for enzymes and catalyzing reactions. Now, I'm sure you must be thinking that so far we've seen enzymes taking part in reactions within the cell. While that is true, this is also true. The enzymes, they happen to be also present in the cell surface membranes and also in the membranes of some organelles. And we're going to talk about that in topic 12 and 13 specifically when we talk about respiration and we talk about photosynthesis. Point number 7, it anchors the cytoskeleton. Yes, we agree in our specification. We're not supposed to be concerned with cytoskeleton so much, but generally speaking, the cell, it's quite a floppy structure. It needs something like a skeleton, and that skeleton is actually cytoskeleton, and that is held in place by the membrane. Point number 8, the cell membrane selects substances that enter or leave the cell. That's the reason why we're calling it as a semi-permeable, or excuse me, partially permeable. Point number 9, it forms hydrogen bonds with water for stability. A very important point for us to understand, because we've done this already in topic number 2, the idea of the water being hydrophilic and hydrophilic actually, because of its polarity. And again, all of these points, the extension of these points is coming in subtopics to come. And then point number 10, finally, it allows cell to change its shape, so it is responsible for the flexibility of the cells. For example, your white blood cells, they engulf the bacteria, and of course that engulfing is done because of the potential of the cell membrane to change its shape. All right, let's proceed. Now, the next slide is basically talking about the basic unit of cell surface membrane, and that is a phospholipid molecule. We talked about this molecule in biological molecules when we were talking about the categorization of lipids. Now, what we need to understand, you can see on your screen, you can see an animated version of a phospholipid molecule. There are two regions. There are two regions that have been identified for you. You have the head, the brown part, is known as the hydrophilic head. And by hydrophilic, I mean it's the polar part. And then you have the hydrophobic tails, the fatty acid tails. They can be saturated, they can be unsaturated. Saturated means, I hope you remember what saturated means. It means that there's no double bond. And for unsaturated, yes, there are double bonds, which causes a slight kink in the fatty acid chain. Okay. Now, excuse me. The first point that we need to know is it forms a bilayer. The cell surface membrane does not exist as, a random arrangement of these phospholipid molecules. No, they're quite well arranged in a bilayer, which I'm going to show you in a minute. Okay. Point number 2, as I said earlier, it has a hydrophilic or a polar phosphate head. So hydrophilic and polar, it's the same thing. And then point number 3, it has a hydrophobic or a non-polar fatty acid tail. Okay, so again, hydrophobic can be equated to the word non-polar, okay? And that's the hydrophilic, that will form the hydrophobic core of the phospholipid bilayer. Point number 4, fatty acid tails may be saturated or unsaturated, as I explained earlier, in terms of the double bonds. Point number 5, now that's very important. I want you to when I say this, I want you to pause the video and really, try and understand and let this point sink in. Polar head forms hydrogen bonds with water. Now, we've seen the structure of water in detail, how it has the polarity. And if you remember, I said water is not the only molecule that's polar. There can be other polar molecules. And polar molecules will be comfortable around polar molecules. Okay, so and that's quite an example for this particular scenario, in which you have the hydrophilic head, which is going to be comfortable. I'm using the word comfortable, of course, it's just a word to explain. But the point is that it will form hydrogen bonds with water, the hydrophilic head. So the hydrophilic head being polar, the water being polar, they form hydrogen bonds. And of course, we understand why that's the case. Point number 6, polar head stabilizes the membrane. Okay, so it's a fluid-like structure. We don't want it to get too much fluid. We need stability. So that's one of the functions of the phospholipid. Point number 7, unsaturated fatty acid tails contribute to the fluidity of the membrane. Now, when I say that the membrane is a fluid structure, we also don't want it to become too rigid, okay? So again, that is kept in its place by the fatty acid tails. And point number 8, these hydrophobic tails are a barrier to the hydrophilic water, soluble substances, polar substances. And that's the reason why anything can't enter the phospholipid bilayer, because of the fact that it has that hydrophobic core in between. Okay, and we're going to talk about that in detail in a minute. Okay, now this is very important. The following diagrams are going to show you hypothetically the way in which a phospholipid molecule will behave on a layer of water. So the first diagram you can see a single phospholipid molecule. I mean, if you have a layer of water and you throw, quote unquote, throw phospholipid molecules, the phosphate head is going to be around the water, and literally the phospholipid molecule will stand upside down, as in the tails being on the upside. And the reason is because of the fact that since water is polar, so is the hydrophilic head. And the hydrophobic tail, which is non-polar, it tends to move away from the water, just because of the fact that it's not hydrophilic. And then again, you can see this is how many phospholipid molecules will form a monolayer. And then most importantly, when mixed with water, in actual they form a bilayer. So what you can see on the right side of the presentation is known as a phospholipid bilayer. That has been highlighted and made bold for you in the text that you can see. Now, again, another diagram which is reinforcing the same concept. When phospholipid molecules, they are added into water, they behave differently depending upon their property. Of course, the property of the head being hydrophilic stays the same. Just the arrangement, it differs. And you can see in all of the diagrams that you can see, either it forms a missile. This circle shape that is formed by these phospholipid molecules is known as a missile, okay? And the bilayer is, of course, it's obvious. Now, again, this diagram is from your textbook. I deliberately put these diagrams just to make sure that you get reinforced when you look at your own diagrams. So you can clearly see that how the phospholipid molecules, they are behaving with water with respect to their nature of being either hydrophilic or hydrophobic. All right, another very good diagram I came across. So you can clearly see it's a phospholipid bilayer on the extreme right. And then in the middle, you have a single layered lipid sphere. And on the left-hand side, you have the lipid bilayer sphere, okay? So you may have water in the middle, okay? And then not in the hydrophobic core, and then on the outside. Okay. Now, of course, whenever we do a topic, it's a good practice to put the concept to test. So that's a past paper question, an MCQ. So let's look at this together. It says when a small quantity of phospholipid is added to a test tube of water and then shaken vigorously, an emulsion is formed by a small droplet called liposomes. Which diagram shows the arrangement of phospholipid molecules in a cross-section of a liposome? Now, I want you to think about this. If you look at this diagram, now let me help you with this diagram. This arrow that you can see that I'm putting, this arrow is pointing towards the fatty acid tail, which is hydrophobic. Okay, that's hydrophobic. Now, we understand that water is hydrophilic. And looking at this diagram, we understand that all around these diagrams, there's water. There's water inside, there's water outside. That's how the question is supposed to be, okay, for all of the diagrams. For all of the diagrams, there's water inside, there's water outside. So considering the properties that we've seen so far, I mean, look at this option. Look at option number A. We understand that this phosphate head can be around water, but these tails to be facing the water, that's fairly impossible because the tails are hydrophobic and they can't form bonds with water, so they can't be around water. So therefore, option A is incorrect. And the same line of argument can be used for B, although in the middle there's water and the phosphate heads, they're surrounding the water. Okay, that's fine. But again, you can clearly see that these fatty acid hydrophobic tails, they can't surround the water. That's impossible. Again, so that's incorrect. If you look at D, again, I mean, this is super incorrect because you can see these tails here, they're facing water. These tails here, again, they're facing water. Both of these tails are hydrophobic and the water is hydrophilic. So again, that's incorrect. So the correct option for us is C. You can clearly see that the phosphate head, it's surrounding the water in the middle and it's surrounding the water on the exterior. And that's exactly how it's supposed to be. You can see the hydrophobic core in between, away from the water. Okay? All right. Now, the following diagram is a two-dimensional diagram of how the phospholipid bilayer would appear. Okay? Now, I know there's a lot of information on the slide. Let's pick up on them one by one. The first thing I want you to know is, in terms of its thickness, the cell surface membrane is 7 nanometers. And this is an assessment, this is from the assessment objective 1, which means that you need to remember this information, okay? All right. Now, to make things simple, when I explain this, I want you to pause the video before I go to the next slide. And there are 5 things that I will identify, okay, that we need to know with regards to the labeling of the cell membrane, okay? Okay. Now, the first thing is, let's start from the, let's start from the left side, and then we're going to go clockwise. So if you look at the extreme left side, you can see this label, the phospholipid bilayer, okay? So they've labeled this entire bilayer as the bilayer because it's being represented by both the phospholipid molecules arranged in such a way. And now we know why this arrangement is in this fashion. And then you can clearly see that they've also labeled one phospholipid molecule. So it's more or less, it's one and the same thing, but you need to make sure that whenever you get to draw or label in the exam, you are clearly able to distinguish between a single phospholipid molecule and a phospholipid bilayer. Okay, so that's number one. That's number one. Let me label that as number 1 for you. Okay, and then number 2, you can see these green fellas. These are the cholesterol, okay? We're going to come at their function in a minute, but you can clearly see that they look similar to the shape of the phospholipid. They're not phospholipids. I just said that their shape is similar. Their head is hydrophilic, just like the phosphate head, and their tail is hydrophobic, just like the tails of the phospholipid molecule, okay? That's number 2. Number 3, you can see that there are different proteins. You can see some of them are small at the bottom side, and some of them are spanning all across the membrane. Some of them have a passageway through them. We're going to come to their categories in a minute, but generally speaking, that's the 3rd thing that we need to know. Now, the 4th thing that we need to know is that I'm circling on this side is you can clearly see that there's a branch that stems out from the phosphate head, okay? Now, this branch, if you like, is a branch of a carbohydrate. Excuse me. Now, since the branch is stemming out from the phospholipid, so you call this thing as the glycolipid. Of course, it has its own function, which we're going to talk about later. So that's the number 4 thing that we need to know. And eventually number 5 is something similar. You can see that again, you have a chain, a spider web, if you like, coming out of the protein. Not the lipid, by the way. This is protein now. So therefore, you call this as the glycoprotein. So that's number 5. So these are the 5 essential labels that you need to know, number 1. Number 2, you need to be able to identify them in a diagram. Number 3, you should be able to draw them. Yes, you heard me correct. You should be able to draw them. I want you to pause the video and try and look at the diagram and count for yourself all of these 5 things. All right, let's proceed. Now again, this diagram is a similar diagram but a slightly 3-dimensional model. The previous diagram was a 2-dimensional version. That's a 3-dimensional model. Again, you can see all of the phospholipid molecules and the bilayer and the proteins, the glycolipids, the glycoproteins. What did I miss? Cholesterol. Yes, that's true. Okay. Now, very important. And let me repeat it again. Very, very important. This model of this diagram that you saw for the cell surface membrane is known as actually the fluid mosaic model. Okay. Now, we need to know, what does the word fluid mosaic mean? Now, the word fluid has a different connotation and the word mosaic has a different connotation. The word fluid implies that the phospholipid molecule that you just saw in the diagram and the protein, I mean, look at this diagram. If you look at this diagram is looking quite fairly stable and you would think that everything is set in its place and nothing moves, but that's not true. I want you to look at this diagram and consider one of the phospholipid molecule, just one molecule. This one molecule, now consider my finger to be one phospholipid molecule. This phospholipid molecule has the ability to move within its monolayer. I mean, this molecule can't go on the top and move at the top layer. So when I say that the phospholipid molecule, it moves within the monolayer, it's the lateral movement that I mean. And number 2, the proteins, some of them are fixed, but some of them are literally floating around this jelly-like structure. So the fact that number 1, the phospholipid can literally laterally move, number 2, the proteins can move and diffuse within the monolayer, this is what is implied to be taken as fluid. Okay? And then you have the word mosaic. That's quite easy to understand. And with no offense to painters out there, but you have people who really are artistic and have a very good aesthetic sense, and they do paintings which literally a common person can't make any sense of. But I mean, they sell those paintings as quite expensive paintings. And the reason is because they have all of those designs which to a common man honestly don't make sense, but they have a very deep implication. Usually those paintings, they are known as the mosaic patterns. So literally the word mosaic will mean patterns and designs. But of course, in our context, what that means is, I mean, again, I want you to look at this phospholipid bilayer. You can see that the proteins, they are scattered all around the cell surface membrane. That's exactly what I mean when I say the word mosaic, okay? So the word mosaic implies different types of protein molecules scattered. Now there's an exam tip I want you to take here. Do not write different patterns. Don't use the word patterns. You know this for sure that the Cambridge, it's quite picky and choosy when it comes to the choice of words, okay? So that's what fluid mosaic means. This is important from the exam perspective. You will find lots and lots of past papers asking this question. All right. Now, some more diagrams. Let's quickly do a run through of some more diagrams which are showing you how a cell surface membrane would look like. Now, this is again a very good diagram in which you can see the blue phosphate heads and then the orange, if I'm not wrong, the fatty acid tails, okay? And then you can see these yellow sticks like things in the diagram, which are actually the cholesterol. And then you can see the proteins. Don't worry about their different names. I'm going to come to that later. And then you see the glycoprotein and you see the glycolipids, okay? And then again, this is the same diagram with its 2-dimensional version given. So it's the same thing. Okay, again, this is a black and white diagram. Again, the point of showing you all of these diagrams is just to make sure that you get reinforced with this idea of knowing how exactly all of the things will be. Now, okay, this is a very interesting picture. It's a hypothetically created animated picture for you to show that if the bilayer was to be separated, how would it look like? Okay, And this idea, by the way, is assessed in some of the MCQs in the past papers. So I really want you to understand and appreciate. By the way, just ignore the E face and the P face. That's none of your concerns. Now you can see if hypothetically we separate, and by the way, this is not hypothetical. There is a procedure known as the freeze fracture in which you can do this, but nevertheless, I'm not going in that detail. The point of this diagram is for you to appreciate the fact that how proteins are embedded within both the monolayers. Sorry, it's basically, the word is skipping my mind. It's basically spanning across the entire membrane, and some of them are just at the boundaries. Okay. Again, this next diagram is a very good two-dimensional representation of all of the five things that we saw. So let's start from the bottom right side, and then let's go anti-clockwise. So you can see the phospholipid molecule, then you can see the glycoprotein, the proteins, and the cholesterol, that little yellow thing at the bottom. Okay. All right. Now again, another diagram in which you can see the upper diagram is actually the upper or the lower view, in which you can see only the blue circles. I want you to pause the video and think about this before I answer this to you. What are these blue things? Yes, you're correct. They're the phosphate heads. And then you can see the entire phospholipid molecule, and these orange fellows are the cholesterol molecules. Now, let's put our so far done concept to test. Let's try and label all of this. Okay, let's start from the top right side, and then we'll go clockwise. So basically, this thing on the top right side that I'm putting an arrow on, this, what do you think is this? Make sure that you pause the video and you think about the answer before I answer. So this is it's a glycolipid. Why is this a glycolipid? Because it is connected to the phospholipid. And then this fellow is the cholesterol. And then this is a protein. It's a specific type of protein. We're going to talk about that. This is also a protein. Again, this is also a glycolipid because it's attached to the lipid, if you look at the diagram clearly. And then this is a glycoprotein, because the carbohydrate portion is attached to the protein. Okay, I hope that was helpful. Now quickly, let's, this is an exam style question. I think it was in one of the recent past papers, if I'm not wrong. Whatever the case is, the question tells you about the fluid mosaic model, and then it says some of the components of the cell surface membrane are phospholipid molecules. That's the first bullet point. Protein molecules and cholesterol molecules. Now, in the box below, draw and label a diagram of a section through a cell surface membrane to show how the above components are organized within the membrane. The diagram should include other named components of membranes. Label the inner and the outer surfaces of a membrane. This is a 5-marker question. I want you to pause the video and do this question. Make sure you don't take more than 5 minutes. Read the question carefully. Draw this with a pencil. That's a question that often students ask. You're supposed to draw the diagrams with a pencil and label them with a pen. Okay, and you don't label the arrows with the arrowheads. Okay, just simple lines. Okay, I want you to pause the video, do this yourself, and then in the next slide, I'm gonna come up with the mark scheme and the examiner report. All right, now, although it's a 5-mark question, but you can clearly see that the mark scheme is enlisting 9 marking points, okay? So the first point is obvious bilayer. So the first thing that the examiner will look is that, has the student drawn the bilayer? Okay, as in literally, so if I help you with the answer, it should be something like this. As in literally, that's how, so this is considered as a bilayer. Okay, so that's what the examiner means when it says obvious bilayer. Okay, with single. Now, of course, when you're doing this, you need to make this habit that you're labeling the hydrophilic and the phobic part. Okay, okay. Look at point number 2: glycoprotein labeled and glycolipid labeled. Okay, so you can refer back to the diagrams, and I'm sure that the diagram that you have drawn, you can clearly see if you've missed out on these. Point number 4: one type of protein drawn. because there are many types of protein that we're going to look at the end of the lesson today, inshallah. So that's what it means. Point number 5, protein type qualified. So the first point is just for knowing proteins. The next marking point is for you to give all of those details. So you can see these names that I'm sure you don't know at the moment, but we're going to come across them. So these are all different types of proteins, okay? And then number 6, you can see cholesterol labeled, okay? And then look at point number 7. You know, you already got the mark for phospholipid, but for marking point number 7 is for you just to tell which part is hydrophilic and which part is hydrophobic. By the way, a hydrophilic part has a separate mark, a hydrophobic core has a separate mark. And then they've also given a mark for cytoskeleton, which your specification is as such not a requirement. If you have able to manage any of these 5 points, you will be able to score the mark. I want you to quickly do a run through of the examiner report. I'm not going to read all of this. I want you to pause the video and read all of this examiner comments for this question. But the point that I want to highlight here is one of the common mistake that students do is, see, I've highlighted this point. It says full credit could only be gained if the diagram was correctly and fully labeled. Now, majority of the students, they did draw the diagram, but they never labeled it. So please make sure, I'm really hoping that you didn't make this mistake. If you did, that's how we learn, okay? All right, now to proceed, cholesterol, it has 3 main functions. Number 1, it stabilizes the membrane. I mean, I want you to think about this. Excuse me. The cholesterol, the membrane is a fluid structure, okay? So, I mean, there is a limit to which it can become fluid. And so there's something that needs to keep a check and balance, and that's basically done by the cholesterol. So it stabilizes the membrane by providing mechanical stability, regulates the fluidity of the membrane by preventing the lateral movement. Remember, I told you that the fact that the fluid mosaic model is called fluid mosaic, and the fluid part means that the phospholipid can move within the model layer. So this is kept in a check and balance by the cholesterol. And the third point is it has a hydrophilic head that I alluded to earlier, and a hydrophobic tail, so prevents the passage of ions and polar molecules through the membrane as the phospholipid bilayer does. Now, this slide is super duper looper important, okay? I wanted to make sure, I'm sure you're taking notes already, but this is going to tell you different proteins and their types, okay? Now, you won't find this in your textbook in this way. I've done the homework for you, so I want you to please make sure that you pay attention. Now, you can see on the diagram, there are 2 different, so you can see the green protein and the yellow protein. They've been labeled as peripheral proteins. And then you can see there's the purple one is the integral protein, okay? So therefore, we say that the proteins, they are categorized into 2 distinct categories. The integral protein, which is also known as the intrinsic protein, and the extrinsic protein, which in this diagram, you can see are the yellow ones and the green ones. Now, the good news for you is, in our Cambridge syllabus, we don't go into much details about extrinsic proteins. We do talk about them in cell signaling, but not in detail. We will go into details of intrinsic proteins. Now, remember, intrinsic proteins are proteins which actually span the entire bilayer, the entire phospholipid bilayer, okay? Okay, so there are 4 types of intrinsic proteins that we're going to talk about. The first one is known as a channel protein. Now, as it's obvious from the name, it acts as a channel for substances to pass through. Now, the following diagram is an animated representation of how a channel protein will look like. So you can see that the ions, they are entering through. So this purple thing is the channel protein, okay? It's a protein channel. And by the way, before I jump onto the carrier protein, let me tell you something about channel protein. You need to remember that they have a fixed shape, and usually they are water-filled pores. And you know, the inner side of the channel protein has the hydrophilic part, and the part that is just next to the phospholipid molecules is the hydrophobic part. I want you to pause the video and think about this, the statement that I just said, and think about the implications. It's a homework for you, okay? All right. And then the next protein is a carrier protein. Again, as it's obvious from the name, it's supposed to carry something. Now, this next diagram is a diagram representing a protein type, which is a carrier protein, okay? Now, it will carry something from outside of the cell to the inside or vice versa. Remember, a clear distinction between a channel protein and a carrier protein is that the carrier protein can change its shape, but the channel protein is unable to change its shape. That's a stark difference between both of them, okay? And by the way, both of them are known as transport proteins. All right, the third category of proteins in the membranes are enzymes, which we know we've talked about how they catalyze reactions. Of course, we don't go into details here, but we understand what that means. And the fourth category is glycoproteins that we just saw. Now, for this fourth category, glycoproteins can have 4 different functions. Number one, either they can act as receptors, they receive something, either a hormone or a neurotransmitter, okay? Or the glycoproteins can act as cell adhesion. Remember when we saw during the start of the lesson when I told you the different functions of the membranes, I talked about cell adhesion. It's actually the glycoprotein that does the adhesion. And then cell signaling, we'll talk about that in detail in the next slide. And then you have antigens. Antigens are basically, they're just the ID cards of the cells. I'm using this example of ID cards because it usually does make sense to students, okay? I mean, think about this example. In a university, every student is obliged to wear their identification cards. And the purpose is that any person from the management can identify that this student is a university student. And any other stranger coming into the campus will have to wear maybe a visitor card, okay? So our cells also have identification cards, and these are proteins which are known as antigens, and they are from the glycoproteins, okay? So please make sure that you really understand all of this categorization. There's another way to understand and to remember actually different proteins and their function. This is an acronym TRACIE, okay, or TRACIE. And T stands for proteins which are responsible for transport. R stands for proteins which are responsible for receptors. A for anchorage. C for cell recognition. I for intracellular joinings. And E for enzyme activity. This is a good slide that I saw on the internet, so I thought of putting it up. I mean, but still, I would rather go for this particular categorization. But this is also a good way you can remember different proteins and their functions. Now, the last thing before we end today is the process of cell signaling, okay? Now, cell signaling, this process is a general outline that I'm going to explain. Excuse me. This can be replicated on different examples in the body, and I will allude to an example at the end. Now, I want you to look at the top left side of the diagram. You can see that green circular thing. It's coming and it's actually going to be attached to that groove on the protein receptor, if you like. So point number one, a signal molecule. So for example, in this case, let's consider it's a hormone. So let's consider that green ball, if you like, is a hormone, and it arrives at the cell surface membrane, and it binds with a specific receptor that may or may not be taken in by the cell. Now, the word specific here is important. I want you to think about this. Consider the name of any hormone. I mean, let's say glucagon comes to my mind or insulin comes to my mind. Every hormone will have a special receptor for it. So for example, a protein which has a recepting site for insulin will not allow glucagon to bind because they're specific in shape, okay? All right, now the next thing is, you can see that the protein receptor has attached to it a peripheral or an extrinsic protein, which is known as a G protein. Now, the second step is that the G protein gets activated and it literally detaches itself from the protein receptor and it moves towards another enzyme, which you can see in the diagram. So point number 3 says G protein attached to the protein receptor moves away from it to activate an enzyme. So the third point, we can see that G protein detaches and it's now activating the enzyme. Now, what does the enzyme do? Once it gets activated, it secretes a chemical called a second messenger, which is a small soluble molecule, and it diffuses through the cell. And you can see this diagram. It's actually these, if you look at these pink things, okay, these are the second messengers. They are activating some enzymes, which are in turn activating some other enzymes. And you can see this literally is a signaling cascade with amplification effect. Okay, so I'm reading point number 4. This enzyme releases a molecule called second messenger, which is small, soluble, and so diffuses through the cell, greatly amplifying the signal. This initiates a signal cascade with amplification at each stage. And at the end, you have the response. Okay, so these are the 4 steps. It's a typical generalized way in which cell signaling works. The following diagram is just an example. You don't need to remember any of these details. I'm just trying to give you an example of that general process. I want you to overlap that into this example. I mean, look at this. That's a hormone. It's coming, it's attaching to a receptor, which activates the G protein. It activates the enzyme, and then you have this cascade of reactions. Okay, so I really hope today's lesson was beneficial. Please make sure that if there's anything that you didn't understand, I want you to go back to the video, reconsider that part of the video, and make sure that the notes that you've taken are really correct. Please take very good care of yourself. Until then, see you next time.